Assalamu alaikum. Hello everyone. My name is Dr. Farhan, and I will be teaching you Cancer Informatics course, and this is a very important course that will help you to understand that how to integrate the information technology into cancer biology and how you can explain and answer a lot of questions you see and you ask using cancer genomics or cancer data, right? So let's begin and let's see what is the importance of this course and why we should focus on cancer informatics, right? So this is a very intriguing or very interesting course for all those students which have a background of biology or biosciences or molecular biology or biochemistry, and they also have a limited knowledge of bioinformatics, and they don't know how to cope with a lot of challenges you see in current science that is related to biology. So I encourage a lot of those students who have the background of biology but don't have bioinformatics to opt this course and follow through the whole lectures. And by the end of this course, I am pretty sure that a lot of the students of the background of biology will learn a lot of information technology, and they can cope with the new and ongoing challenges in cancer genomics, right? So why we should focus on cancer informatics? Let's answer this question. So first of all what we need, all of us know that cancer is very challenging and a very complex disease, and it's very simple to say that we are looking for the cure of cancer, or we are trying to find the different genes which have association with cancer. It looks very simple. It's like we say that if, for example, there is a gene called any gene, you can say, if I just write it the name, for instance, I call it, there is any gene like TP53. Now we know that TP53. Now we know that TP53 gene is actually a tumor suppressor gene. I'll go through the basics in the next slides, but now we know that TP53 is a gene which causes, this is known as tumor suppressor gene, and this causes a lot of different diseases if there is a dysregulation in this gene. So if we try to find any mutation or any problem in this gene, so we can say that now we have identified that this particular gene is causing cancer. But it looks very straight and simple forward answer, but this is not the case. There are a lot of things which we have to consider when we are talking about identifying the genes, because as the lecture goes on, you can see that there are so many factors, there are so many features within a genome which you have to consider while looking for the cure, or while looking for the diagnosis of the cancer, or while understanding the prognosis of the cancer. So first of all, here you can see that cancer refers to more than 100 forms of disease. So what does that mean? That there are every tissue of the body, every cell and every tissue has different type of tumors. If you name the cancer tumor tissue, that tissue may have some certain type of cancer. So it means that when we are talking about the cancer, it can be of any organ or any tissue, and the problem integrates. Sorry, not integrates. The problem aggregates when we see that the the actually the features of each cancer type are different. Like, if for example, if we are talking about liver cancer, then the features or the factors which are associated with liver cancer will be different. When we are talking about lung cancer, then there will be different factors associated with the lung cancer. But again, there are some similarities as well. So we have to draw a thin line between different cancers, and we also have to combine all the cancers and find the common cure. So there are a lot of things which has to be addressed in cancer genomics, and that cannot be addressed without the information technology, because information technology provides us with the platform. So to find the easiest, or you can say ways to organize the data and then find the answers. So again, about the cancer, as I've written here, that in simple words, normal cells are all alike. but every cancer cell is cancerous in its own way. Like for example, this is a very famous proverb, or you can say quote of Leo Tolstoy, that happy families are all alike, but every unhappy family is unhappy in its own way. So we are talking about unhappy families or unhappy cells, that each cell may if each cell which has any problem or which has any tumor will have a different characteristics altogether. So trying to classify those different features for each cancer is difficult. And again, and the problem again aggregates more when we are talking, for instance, we are talking about liver cancer. So there are different types of liver cancer. For instance, if we are talking about there are certain type of patients whose phenotypes are associated with some viral infection, some are without viral infection, some are due to alcohol. So all the environmental factors will be different, and then the genomic factors will also be different. So these are a lot of challenges which we have to understand, and that understanding can be done using cancer informatics. So next, if here you can see, this is just a slide from um, worldwide cancer data. You can see that, as I said to you, that this is just a list of 12 cancers. If you go on this website, the link is available, and you can see that this link, you can go to this link, and you can see that there is a list of cancer, and all possible tissue types have different cancer type. And again, you can see that each cancer, the incidence of each cancer is very high. For example, the most common cancer around the world is lung cancer, then prostate cancer, colorectal cancer, stomach, liver, and so on and so forth. And again, then the complexity, for example, some cancers are more deadly, or you can say more aggressive, and they give poor phenotypes compared to others, but still cancer has its own characteristics which we have to address, right? This is a very famous article which was published in 1997. It's very old, but it's very relevant here because New England Journal of Medicine, this is the top journal of in the field of biology, with an impact factor of more than 50 or 60, and they actually analyzed the data from starting from 1986 to 1994. You can see that they collected the data from 1986 to 1984, and they tried to identify the trend in mortality due to cancer in the United States. And because a lot of research started in 20th century, and that was getting faster and faster when 20th century was, when we are nearing towards the 21st century, and they tried to analyze that what is the outcome of this research, and whether the mortality rate of the cancer is decreasing. I think you know what does this mortality means. This means the death rate. The death rate of the cancer is decreasing or not. And surprisingly, they identified that as the time progresses, the mortality was increasing, but that was somehow decreased in 1994, but from 1992 to 1994. But here you can see that from 1986 to 87, 88, 89, 90, 91, the mortality rate was increasing. So they were very concerned about why this is happening, that the cancer, rather than we try to inhibit the cancer, rather than we try to stop the cancer, here we are actually losing the battle, and why the cancer is undefeated. So I'll share with you this article, and you can read it out, that there are a lot of things which were lacking in the research. And what I understand from this as a bioinformatician is that we are not looking at cancer in totality. Like for instance, we are not looking into why this is, how the genome of the cancer is behaving. We were making small targets, and we were trying to achieve the targets. For instance, we were looking for some genes, some regions, and some phenotypes, but we are not looking at all the features, all the genes, and whole genome at a time. So this was the biggest problem that the cancer scientists were facing at that time. And now, why that cancer was undefeated? There are a lot of reasons. For instance, there are a lot of conspiracy theories. We know that some people say that if we just this is just for making money, this is for the business, or some say that if we just give vitamins and the cancer will be cured. We are not going into those we are not going into those issues that why there are a lot of conspiracy theories, but we will, because that is beyond the scope of this course. We are actually looking into the reasons why the cancer, real reasons why the cancer is undefeated, and our objective is to look into the whole genomic data, whole proteomic data, metabolomic data, metagenomics data, and transcriptomic data. And these all sort, if you know the terminologies, for instance, if we can call it in common, if we take the common, we can call it the omics. For example, omics include genomics. Genomics means the study of the genome. Genome means that all the genes present in the genome at the DNA level, whole intronic or exonic regions and genomic regions that we study, that is called genomics. Again, if you're talking about proteome means that we are talking about the whole proteome of the genome. That is actually all the proteins in a single individual. Again, metabolomes, if we are talking about all the metabolome, and then metagenomics, and then the transcriptome, if you are talking about the whole transcriptome, all the genes, all the RNAs present in one single individual. So throughout this course, we will be looking into the genomes, proteomes, and transcriptomes, because we are dealing with the large data, and I will explain you how this genomes, metagenomes, proteomes, and transcriptomes are important. And in current time, for example, we are talking about 2021. Right now, this is very doable. This is very easy. This can be done. Not There are certain levels. For example, if you are a biologist, again, if you're a biologist, then there are a lot of ways you can analyze the genomic data. There are a lot of ways you can analyze the proteomics data. And if you are a bioinformatician, then it's a plus point, and you can further make different ways of interpreting the data, integrating the data. For example, if we are a single liver cancer genome, right? Now, if we are talking about liver cancer genome, we can study the genomic that individual at a genomic level, then the proteomic level, and then the transcriptomic level, and then we can compare that how this liver cancer originated in this individual. And then if we increase the patient information, for example, first we have one liver cancer patient, and then now we are talking about multiple liver cancer patients, and then we can find the common features in the genome or the transcriptome or the proteome, and then we can come up with the new ideas that how this whole omic picture is behaving when we are talking about the cancer, right? So there are a lot of things that can be done in cancer informatics, and this all comes under the umbrella of cancer informatics, right? And again, why this is so, again, I've talked you through that everything is about the genome, and the genome is important, and we will study the genome, and we can study the omics. So if we look into this article that was published by Michael Stratton in Nature in 2009, and he showed a very beautiful picture that how the cancer genome actually behaves, or you can say how the cancer is linked with the genomics, right? For instance, here you can see that it's talking about starting from the fertilized egg to it's moving towards the gestation, infancy, childhood, adulthood, then early clonal expansion, benign tumor, early invasive cancer, late invasive cancer, and then chemotherapy resistant recurrence. So these are different terminologies we will go through. I'll share with you a lot of articles. You can go through all the articles, and then we can discuss everything during the question answer sessions, and you can come up with the new ideas, and I can I can discuss them, all of those with you. So here you can see that here it's talking about starting from fertilized egg to chemotherapy resistance recurrence. So let's see that the slide shows you that, for example, at all the mutations occur within a genome at different levels. For example, yeah, here you can see that in the gestation stage, for example, there is one mutation, that is intrinsic mutation, that occurred, right? Mutation means a single nucleotide change. I'll go through the details later on. And again, then those mutations or changes become frequent with time. For example, there was one mutation at gestation level, then there are two mutations at infancy level, then there are some mutations of different characteristics at in childhood, and then adulthood, and so on and so forth. The mutation level is increasing exponentially, right? So then there are different types of mutations. Here you can see that circle mutations are known as the passenger mutations. I'll go through the detail again later on, but here for you, that passenger mutations are those mutations which do not show any substantial change at the genomic level, so there is no significant change in the genome. But there are some sort of mutations that are called driver mutations, and those driver mutations actually change the structure of the gene and then disrupt the whole genomic function, and that genomic function is obviously linked with some protein function, and then the pathways and all the linkage of the gene and protein and the pathway is disrupted due to some driver mutation, and that causes a tumor. So here you can see that at this point of time there was first mutation at that is known as early clonal expansion, and this driver mutation actually generated a benign tumor, and that tumor is then the building block to initiate the cancer. And here you can see that after the benign tumor there were other driver mutations that took place, like this one, and the tumor expanded, and you can say the tumor became invasive, and then from invasive that made it further becomes a shows for aggressive phenotype, and from benign to invasive, and then the metastasis occurs. And then there are a lot of, again, a big challenge is that so we know that there is a way to cure the cancer, to minimize the cancer, is that we do chemotherapies and radiotherapies, but there are a lot of mutations which actually are also resistant to chemotherapies. So there are so many things we will discuss one by one in our class during the course, and we will discuss that how to classify that these are the passenger mutations, how to classify these are the driver mutations, how to classify that these mutations are chemotherapy or any treatment resistant. So these all things will be done, or you can say will be discussed during the course, and that will help you to do your own analysis with your genomes or data of interest, right? And why? Yeah, and one more thing, that why this whole data is very complicated, and it's a lot of data. That if you talk about a genome, any genome, right? That genome is the size of that genome is 3.2 gigabases, right? And this 3.2 is actually this whole big size. There's one base pair file, 1 KB file, 1 megabyte file, and then gigabyte. And this is a very big file, and that is of text file which is 3.2 GB. And when we classify that information, we can see that there are a lot of information that out of this 3.2, only 1.1% is the exon, 24% are the introns, 75% are the intergenic regions. That intergenic regions means that the regions between the two genes. So all this information is present in the human genome, and if we are talking about cancer genomes, and we want to find a cure of any specific cancer, then we have to look into all these exons, all these introns, all these intergenic regions, and try to find what are the important features or what are the important regions which are actually causing the cancer. And this is a very big challenge. If we are if or it's a very, you can say it's a big challenge, or it's a big field, interesting field, where you can play around with a lot of data and you can come up with a lot of ideas, rather than looking for a single gene or two three genes and then finding the association, because all of these things are interrelated. For example, here we are talking about exons. So exons are actually those regions which code for a gene, right? And that gene actually produces a, or translates into a protein, and that protein has a specific function. So if we are looking for any specific genes, we are looking for the expression of those genes, or we are looking for the protein phosphorylation or anything, we will be focusing on these regions, right? And 1.1% exonic regions. But if we are looking for other regions, for instance, there are a lot of regions, intronic and intergenic regions, which regulates these exonic regions. Like we can say that there are some promoters, enhancers. Enhancers are those which enhance the expression of a gene. Then there are silencers which inhibit the expression of genes. Then there are transposable elements, and so on and so forth. There are a lot of things which are present in the genome and the intronic regions or intergenic regions. Then again, micro or non-coding RNAs. So if we are, which actually regulates these genes. So if we want to look for a certain gene, for example, we are looking for a gene like CDK1, and now we want to see that the expression of this gene is increased in cancer, right? Now we want to evaluate how the expression was increased, whether there are some enhancer regions, whether there are some present in the introns or intergenic regions, or any other in the exonic regions. So we have to look for all the data, and we have to come up with the answers of those questions, that how these intronic or intergenic regions are regulating the exons. So at a time when we are talking about the genomic data, we have to look in totality and try to find the network, that how any specific gene or any specific pathway or any set of genes are behaving, or they are causing any cancer, right? So that for that we have, if we want to categorize cancer genomes efficiently, we have to look for the whole data, right? Next, and just for your information, this is a human genome general features that was downloaded from Ensemble Genome browser, and you can see that it's a bit older, but the information is would not be substantially changed. Here you can see that the human genome was sequenced using a shotgun method. This is not the I'll explain later what is the shotgun assembly method, but the whole genome was sequenced using this method. Then there are a lot of 27,478 contigs identified in the genome. Then 3.2 billion base pair consensus sequence. 3.2 billion base pairs. It's a huge number of the consensus sequence. And then using some tools, the genes are actually the coding regions, or the protein coding genes that were identified or predicted using a gene scan. This is one tool which we may use later on, that predicted that there are 43,887 protein coding genes, and then the known protein coding genes which are actually experimentally validated, and which are the key genes which are known as well, are 22,286. This number changes as the science progresses, but commonly we say 23,000 protein coding genes. Then there are 12,308 pseudo genes. Pseudo means false genes that actually looks like a gene, but they are not gene. For example, maybe due to some mutation or any evolutionary change in the gene coding region, now the gene is non-functional, but that is present as a gene in the genome. So there are 12,000 of them. Then RNA genes, there are almost 10,000 RNA genes, which mean that there are a lot of non-coding regions microRNA genes, single small RNA genes, and so on and so forth. We will discuss this later on. Then there were single nucleotide polymorphisms. Again, I will discuss this later on, that there are 1798125 SNPs were reported in the genome. So if you look into the data, this is a huge information. And for example, there is some this genome is diagnosed with cancer, and now we want to find the cure. How will you find the cure? If we are looking for a one gene or two gene or three genes, we have to look for everything here. And this is not complete, as I showed you in the previous slide, that there are actually 99% of the genome that is not the coding part, that is not the RNA part, or so on and so forth. So this is only the 1% of the genome. But if we want to look in totality, how can we find the cure? The best way is to manage or analyze the whole data using some cancer informatics or genomics techniques and tools, and we come up with the answers, right? So now you may understand that why cancer informatics is important, because information technology plays a key role in fighting cancer. You should understand this, because without the information technology, without your computational techniques and computational approaches, you cannot fight with the cancer. Because there are a lot of things which are still largely unknown, because of the challenge in maintaining, analyzing the data. So what is basically the definition of cancer informatics here? Cancer informatics is where information science, computer science, and healthcare intersect, right? So healthcare, again, it's about acquiring, storing, and using information about cancer most thoroughly and efficiently. This is somewhat related to the definition of bioinformatics, as you may have studied that bioinformatics is about acquiring, storing, managing, interpreting a large amount of biological data. So here we are talking about acquiring, storing, and the information about cancer thoroughly and efficiently. So thoroughly is very important, and if we are looking thoroughly using some efficient techniques, then we can come up with a lot of new ideas, a lot of new ways to fight cancer. And cancer informatics is a broad field, getting deeper and more complex as medical research and information technology progress. Yes, that is true. That as I showed you here, that as the information of cancer genome is increasing, as the new genes are identified, as the new phenotypes are identified, as the new environmental factors are coming our ways, there are new ways and there are new challenges in cancer informatics to deal with the data. And that is what we do in cancer informatics. So the field is all about collaboration, not just among physicians and researchers, but between doctors and health IT professionals. This is very important, because as a cancer informatician, or you can say as a bioinformatician, we are not capable as the clinicians. We are not capable as the healthcare officials. We are not capable as the pharmacists. We are not capable as the pathologists and the medical doctors, and so on and so forth. So what we have to do here is that cancer information is all about, why it's all about collaboration, because what we can do as a informatician is that we can handle the data, we can analyze the data, we can organize the data, we can come up with the patterns that these genes, set of genes are behaving like this. But again, we need a lot of input from the health IT professionals, we need a lot of input from the clinicians, from the pharmacists, from the doctors, that okay, if this gene is behaving like this, and then the clinicians will give us the input that the tumor is progressing more aggressively in these type of cancers. So we can integrate all the information, and we can make a link between the cancer biology and information, and we can come up with the new solutions, and we can validate those using new cases, and we can check whether the pattern we identified is consistent in other cancers or other patients as well or not. So that's the take home message of this lecture, that is, cancer informatics needs to be known. Cancer informatics has to be used when we are talking about when we are talking about finding the cure of cancer, fighting the cancer, because without cancer informatics, only the clinicians cannot find the cure, because because they don't have the information, or they don't have the critical knowledge of information technology. So when we collaborate across the fields, then we can come up with the new ideas. And in this course, we will discuss that how number one, we will discuss that how cancer informatics is progressing. Number two, how you can use that cancer informatics information and integrate into your research. And then and also we can also use some different strategies or different methods or tools or languages to understand how to analyze the data. For example, the best thing is that if I give you the patient information, like here you can say I give you 100-200 MB file of any cancer patient, and I'll ask you to analyze this and find the best way of extracting the important information out of all the information. Like if there are 22,000 proteins or 22,000 protein coding genes, I'll ask you find the most recurrent or most important genes which are actually behaving differently in this cancer. So you will hopefully, inshallah, learn this during the course. And this will be a very interactive course. I will try to ask as many questions as possible. I will give you things to perform. I will ask, hopefully we will also discuss different things during the presentations. And yes, I hope this course will be a worthwhile addition to your knowledge, and I hope that I also learned from you. Thank you. Bye bye.